Now that you have a better sense for the types of enrichment analysis approaches we'll be using and that you have available to you, we're ready to start the Step 7 script. Before we jump into Step 7, I'm going to just return to Step 6. Make sure you're looking at the Essentials code chunk because I just want to highlight there's very little software that you needed to get through that Step 6 script to get where we need to be for today. We talked a lot, probably more than you wanted, about colors. We carried out an unsupervised clustering of our rows and columns, which gave us a chance to talk about clustering methods in general, hierarchical clustering, as well as other unsupervised methods, appropriate correlation approaches like Pearson and Spearman, and why we use one or the other. We then talked about how to move from our clustering result to produce modules by cutting our hierarchical clustering tree. And then we produced a heat map. And from there, the rest of the stuff was about how to slice our modules out of that heat map and to do other kinds of manipulations of the heat map, like averaging replicates to produce only a single column in the heat map per replicate or per group, how to plot an a priori heat map just by reading in a data frame or data matrix and turning the numeric data into a heat map. And of course, we talked about the importance of understanding what we're doing when we're scaling the heat map. Okay, so with that in mind, we're now moving over to the Step 7 script, and we're going to do what we always do. We're going to go Edit, Folding, Collapse All to get this high-level view. We'll talk about the script, but briefly, because you already have a sense of what we're doing here. We'll look at the packages we're going to load, and then we're going to carry out gene ontology enrichment analysis using gprofiler2, an R package that will let us access the gprofiler web service. We'll then carry out gene set enrichment analysis using the cluster profiler package. If you recall from the slides, this is an R package that lets you tap into a whole bunch of different enrichment analysis approaches, but also different graphics. It's really got a lot to work with, and we'll use just a few of the functions from Cluster Profiler. We'll then return to our venerable sort of favorite R package, Lima, because as I mentioned before, Lima is quite sophisticated, has a number of different functions for carrying out all kinds of different aspects of gene expression data analysis, including functions for carrying out GSEA. So we will talk about a competitive gene set enrichment analysis approach called Camera that really is one of the only tools that tries to cope with or deal with or address this intergene correlation issue that I raised earlier that can inflate your type 1 error when you're doing GSEA. We'll also use an example of a self-contained gene set enrichment analysis approach called ROAST. In comparing the results we get with Camera and with ROAST with a sort of mock data set, you'll see why and how those underlying null hypothesis that each of these addresses is different, as I described in the slides, and how that plays out in the results. We'll then carry out, I think, a pretty unique and sometimes very useful way of running GSEA, where we don't just compare groups, but we look at variation in our enrichment signal across all our samples. And then, of course, we have the Essentials code chunk. So that's what we're looking at for today. So let's get started. You already understand why we're actually going to be, what we're going to be doing in this script. But I want to just highlight that there's a few things you should be coming into this script with, and they should already be in your environment. If they're not, you'll want to go back and generate them. The first is we want that normalized filtered expression data. This is the data set that is a matrix with, symbols, gene symbols as row names or transcripts, and that's what we generated several steps ago in our R code, and you'll see the object and its name in just a little bit. Of course, you need your study design file. We'll want that contrast matrix that we set up before we did differential gene expression testing, or when we were getting ready to do it, because that actually lays out the pairwise comparisons that we will want to test. And that becomes useful, particularly when we use the Lima-based approaches of Camera and ROAST. And then finally, we need signatures, or even whole collections of signatures. Those, again, could be things you custom make, but we're, of course, going to take advantage of MSigDB and directly access collections of signatures from MSigDB. And so we're ready to go. The packages that we're going to load, some you've already loaded and downloaded and have worked with, Tidyverse and Lima. You'll want to go ahead, if you haven't already, you'll pause the video now and go ahead and load those. We'll also be working with gplots again, which we use to make heat maps, DT, but the rest of the packages below DT are going to be new for you. We're going to be working with GSEA base because it contains a lot of basic functions for doing gene set enrichment analysis. Biobase, which is probably already loaded or downloaded and often loaded by some of the other packages we use. GSVA for carrying out that gene set variation analysis that we'll do in the last part of the script. gprofiler, cluster profiler, msig dbeer. So as you can imagine from the name of this, it provides access to the msig database of signatures, of collections of signatures. And then enrichplot for making a number of different graphical representations of the results from our GSEA analysis. All right. Once you've got all these loaded, which in my case I've already loaded these in the R environment, so I think I'm ready to go. You're ready for the first real analysis approach here. So we're going to carry out Go enrichment using gprofiler, and gprofiler is also, as I mentioned, a web resource that you could use to carry out enrichment analyses directly online. And it has some nice graphical, or at least I think they're really nice graphical representations of the results, but I find it easier, faster, and more customizable to just do it directly within R. So what do we need? Well, it's a Go enrichment. So if you remember from the slide presentation, you need to have selected a set of genes out of the universe of total possible genes in your data. So we've already actually done that, and we did that in the past using this handy top table function from Lima, where we would use our Bayesian stats from our linear model fit. We would adjust for multiple testing using Benjamini-Hochberg. We'd pick the pairwise comparison in our data. In our case, we only have one, so it's going to be the first one that you want to identify genes that are most differentially expressed between. And then we pick the number of top genes we want sorted by one of the statistics in the table. When I run that line of command, and you've done this before, although this number has been changed several times depending on what we were doing. Remember, we raised this number really high to make our volcano plot. But when we run with the top 50, we've now picked a set of genes. We have the genes that are the most differentially expressed between samples from patients with disease and healthy controls, and sorted by their log fold change. So matrix metalloprotease seems to always top the list here, MMP1, at a log 2 fold change of 10, so huge fold changes. Once I have a set of genes, and remember, I don't need the data, okay? The data was used to produce this set or arrive at it. Now that I have it, I just need the row names. This is a data matrix, so these gene symbols are row names. So any data, any set of genes you have can now be used for what I'm about to do. I use top table to arrive at it, but you could use whatever you want. You just have to provide a vector of identifiers. In this case, row names of my top hits is a vector of length 50, a character vector of length 50. All of them are gene symbols. I then just have to indicate exactly the organism I'm working with and the kind of correction for multiple testing that I'd like to do. Those three entities, your character vector of identifiers, your organism of interest, and the correction method, I'm going to pass those to the function ghost, and this is coming from that gprofiler2 package. When I run ghost, which I'll do now, it's running a Go enrichment analysis comparing the genes in my set to all the genes annotated in humans. And what it's doing is it's looking for any Go CC, Go BP, or Go MF terms that are overrepresented. This is that overrepresentation analysis, an ORA analysis, frequently done using a hypergeometric test or a Fisher's exact test. It's using that underlying ORA analysis to figure out whether in the world of possible functional annotations on all genes, whether I ended up with ones that are enriched or are observed more than would have been expected by chance. All right, so I end up with this ghost res, which is a list object, and that list object contains a data frame with a bunch of other information in it and some metadata about it as well. Doesn't really matter this list object and what's in it. We're not actually going to access any bits of that. We're going to pass that, and this function is called ghost. We're going to pass that ghost result to another function from the gprofiler2 package called ghostplot. And it's going to generate the exact same graphic, interactive graphic, that you would have gotten on the gprofiler website. I'm going to pass to ghostplot the ghost result. I'm going to tell it I want that plot to be interactive, and I'm going to tell it I want it to cap, and you'll see what this means in just a minute. All right, so we're running this. It's going to appear in our viewer tab because, again, it's an interactive plot. And when it comes up, we're going to discuss how to change this, how to customize it, etc. Okay, so we're looking at all the gene ontology terms that are enriched in this set of 50 top genes picked in my data set. And the first thing I would just emphasize is what we're looking at here, and what I find appealing about this is it's a Manhattan plot. You can call it a ghost plot if you want, but it's really just a Manhattan plot where significance is indicated on the y-axis and categories or groups of types of functional annotations are grouped on the x-axis. So go MF, go CC, go BP. You know what those are. But the ability to carry out an enrichment or an overrepresentation analysis is in no way limited to Go terms. In fact, any annotations, functional annotations you could attach to a gene can be the basis for carrying out an overrepresentation analysis. For example, you could look at protein domains. Right? We can take any given gene, you could annotate it with predicted PFAM domains, and then you could ask, in the set of genes I pulled out, do I see certain PFAM domains, like DNA binding domains, overrepresented, right? So that kind of generic approach is really powerful, really flexible, and easy to generalize. So that's why we have additional categories down here. KEGG pathways, reactome, transcription factors, microRNAs, I don't know what HPA or quorum are, and additional information here. So mousing over this, because it's interactive, lets me immediately see the Go term, which is right here at the top, and the name of the Go term, immunoglobulin receptor binding, and its p-value. All right, and this is an adjusted p-value. It's a negative log 10 of the adjusted p-value. So I'm looking at FDRs here. This is interactive, so I could zoom in if I wanted to. Not that I need to, and I can go back, return to the original axis settings. You notice how anything with a negative log 10 adjusted p-value greater than 16 hit a ceiling here. That's because I've capped. If I really want this to disperse, it'll make it hard to see some of these down here, but I could set this to false and rerun this plot. And now I'll no longer cap this. Things will display on the axis as high as they go. So you can sort of get some of these to spread out a bit more at the expense of some of the lower things. So now we've got a bunch of stuff crunched down here, but we can see that this immunoglobulin complex Go term is the most enriched in terms of significance. All right, complement activation, humoral response, antigen binding. So clearly there's an inflammatory response, and one that seems to have a ton of B cell qualities or functional annotations associated with it in this data set from at least the subset we're working with. All right, so I'm going to go back to capping that as true. And I'm also now going to set interactive to false. And I'm going to show you what this looks like. And instead of showing in the viewer, of course, it's going to pop up in the plots tab, at least I think. Yes, there it is. So no mouse over anymore. Now this could be exported and used in as the basis for a figure. Now, if I were to save this, let's say assign it to an object called my ghost plot. All right, that's now stored, this image. I can now annotate it a bit more using a function called publish ghost plot, which takes the plot you just produced, has to be static. It can't be interactive, obviously. And you can start to highlight individual Go terms. So I picked one out of here that I saw when I moused over. And now if I run this, it will produce that same ghost plot, static, but it will annotate it with the individual Go term that I've picked out. This might be useful for you. This is also the kind of thing that you could obviously do manually. So it picked out, it circled it, put a 1 by it, and it indicates the source, Go MF, the actual Go term ID, its name, how many genes had that term, the term size, and then the p-value. And of course, this is the false discovery corrected p-value. Okay, so I think now you have a sense for how to quickly do Go enrichment in a way that produces a visually compelling graphic that lets you sort of peruse the results in an interactive way, but then can quickly turn that into a static graphic that you can use in a paper. Now you can repeat this same analysis using one of our modules, right? The whole point of clustering and making a module was that we were going to pass those modules to things like these kinds of Go enrichment tools. So instead of my top hits, I could do my module. And if you remember, my module was, I think, all the either upregulated or downregulated genes, depends on what the last bit of code you ran was. But that my module can now be used in just the same way as my top hits. And so here it is. Here's my module. It's a data matrix as well. It has to be because it came out of making a heat map. So I can capture the row names of that. It's just going to be a character vector. In this case, a much longer one. It's not 50 genes. It's hundreds of genes, over 1,000. I'm going to pass that to Ghost, tell it it's human, and to false discovery correct. It's going to run that now. All right, while that's running, the plan now, and actually I don't need to save that. Let's actually go ahead and produce an interactive plot. Whoops, sorry, I do want to do that. Let's produce an interactive plot here and have it just display directly. And I'm just going to clear off this interactive here and clear this. And we're ready to plot this now. Okay, so now when this displays, I'll be looking at the Go enrichment results for just one of those modules that I identified in the step 6 script. So if you have for your own data a ton of different modules, this is a way to iteratively go through them and really explore the functional enrichment results you get from those. So if you're not a fan of looking at GoMF or GoCC, you can keep your eyes focused on some of these. We can clearly see there's a ton of immune, interferon gamma, granulocyte regulation, etc. So if looking below just those 50 genes and taking a module of coordinately expressed genes shows me something really quite different. It's not just immunoglobulin, B cell defense. It's actually a ton of T cell inflammatory stuff at the top of this list. In fact, allograft rejection, which has driven by T cell responses, is one of the highest results from the KEGG pathway, or the highest result. All right, so again, this is a great way to kind of go back, and I suspect, although I won't do it here, I'll leave it to you to do, if we look at the module of genes downregulated between patients and controls, we'll see a very different functional profile. All right, now you know how to run Go enrichment. We will leave that bit there, and I will start again with GSEA in the next video.